Hello everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklakov at the University of East Anglia, and I'll be your host for today's session. Myself and our wonderful team of co-organisers, Andreas, Ulia and Wouter, in the groups of Judith Mank and Simone Imler at the Universities of British Columbia and of East Anglia, are very grateful for the enthusiasm from the research community for this initiative, and we've been honoured to host a variety of fascinating speakers so far. So thank you all for your fantastic engagement in the series. Before I introduce today's speaker, I'd like to remind you that there will be a Q&A session directly after the talk. So please post your questions in the dedicated Slack channel and upvote questions you'd like to hear. We aim to keep the whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Professor Thorsten Reusch, Chair of the Marine Evolutionary Ecology Research Unit at the Geomar Center for Ocean Research in Kiel, Germany, and previously Professor of Plant Evolutionary Ecology at the University of Münster. Thorsten's research explores the evolutionary responses of marine populations to global environmental change, such as the warming and acidification of oceans, overfishing, and biological invasions. He uses whole genome sequencing techniques across a range of species, including cod, coral, and seagrasses. Thorsten's recent research is unraveling the mysteries of why asexual reproduction can evolve, and how the evolution of male pregnancy in pipefish, seahorses, and sea dragons can inform our understanding of immune system flexibility. Today, Thorsten will present his talk entitled Clonal Reproduction is Not an Evolutionary Dead End. So Thorsten, thank you for accepting our invitation, and over to you.
Yeah, thank you very much, Elizabeth, for a very nice introduction. And actually, I'm very enthusiastic to talk during this really great venue of the online worldwide evolution and ecology seminars. And so the maybe first 1 or 2 words on the Geomar itself. So we are supposed to be the German Ocean Institute. We have grown pretty large, so we are now 1,000 employees and students altogether. And our mission is to study the ocean, so all aspects of the ocean system in terms of natural sciences, from the deep sea to the atmosphere, in terms of geology, biology, biogeochemistry, and physical oceanography. And as Elizabeth has already nicely introduced, so one of the main topics of the biology subgroup is to study the responses of marine life to ocean change. And so why is clonal reproduction and ocean change an interesting question, an interesting bridge to make? So first of all, the background is that clonal reproduction, that often goes hand in hand with modularity in multicellular species, is extremely widespread life history. So it's estimated that 60% of all plants, the majority of fungi, and 40% of animal phyla do clonal reproduction. And I do not mean here asexual reproduction of microbes. That's a completely different story. Here, rapid evolution, including, for example, antibiotic resistance evolution, is widely accepted. What I mean here is budding or fission or branching in multicellular animals, fungi, or plants. It's important that we know what we are talking about. So a very useful terminology was introduced by John F. Harper in 1977 in its seminal book, The Population Biology of Plants. So if you have a growing clone, be it a plant or also an animal, so also for corals, this terminology has been adopted by many groups. You have the module called the ramet that is potentially or de facto independent, and very often it becomes independent naturally after a short time, because the connections here in the case of the seagrass, they are naturally rotting away after a few years. And the whole clone we also can call a genet. That's ultimately the result of a sexual recombination event. And how many clones sit in a certain area has important consequences. So in the wake of the biodiversity ecosystem function debate, One of the interesting questions to resolve, or the riddles to resolve, was that in very species poor communities, or I should say that are species poor with respect to the foundation species like seagrasses, so how can diversity play out as a foundation for the entire ecosystem? And here it turned out that both in marine plants, so the study object of today, the seagrass, but also in terrestrial plants such as this goldenrod, we find that the number of plant genotypes, or genets, we should say, according to the just introduced terminology, determines the dependent arthropod richness in terms of the goldenrod, or other faunal abundance in terms of the seagrass. So this clonal or genet diversity per area replaces species diversity. And that was kind of nice because in a way it unified the biodiversity ecosystem function relationships from the species diversity down to the genotypic richness diversity. However, with genetic marker in the past 10 or 15 years, it turned out more and more that we have situations, and they are really not oddities, they can be very widespread. We have situations where a seagrass bed is a single clone, or a coral reef is really dominated by a few clones that get hundreds of years old. And evolutionary textbooks either totally ignore the question of whether clones can evolve. They basically, if they say anything about the generation of genetic variation, they basically state that asexuality is a special case, and that would just leave genetically identical descendants, period. And this question has fascinated me now for about 20 years, since we discovered some of those seagrass megaclones in the Baltic Sea, but later also at the southern edge in Portugal. So how is it possible that these large clones, or genets, sorry for the typo here, with essentially zero genetic diversity, how is it possible that they have persisted over hundreds to thousands of years? And one of the hypotheses that is then always taken but rarely, seldom tested, is that these may be those genotypes that just hang out there after this enormous selective filter of varying environmental conditions. So these are just the remnants, the most plastic ones that survived. I find personally that hypothesis very unsatisfactory, and the key theme of my talk today is that it may be that the premise that those modules within the clones have zero genetic diversity is actually wrong. And to test this, we went back to one of those clonal seagrass beds in Finland that was done with my close and longstanding collaborator, Chris Boström. And we went to one of those seagrass beds where we already knew from microsatellite marker studies that there's something going on, because we quite often found pseudo triploid patterns that we interpreted as genetic mosaicism, meaning that we have a mix of somatic cells showing a different microsatellite genotypes, and if some of the alleles are non-overlapping, you would get 3 or sometimes even 4 allelic peaks at one locus. And this is even although we can exclude that we have a polyploidic situation in these plants. And we also were fortunate that a few years before, in a large collaborative effort, where we also were one of the lead labs, we were able to sequence the genome of Zostra marina, of that eelgrass growing in the Baltic Sea. And we found out that it was a rather compact genome with a little bit over 200 megabase pairs, which made it actually a very nice study organism to get some decent genome coverage to go after the possible somatic genetic polymorphism within one of those clones. And also that backbone genome site was very close by, so that it was a perfect mapping backbone genome to do our allelic calls in the target genotypes. And in fact, it was the case that we found pretty remarkable levels of among ramet polymorphism. So let me first introduce here Lei Yu. So he's a PhD student in our group, and he did the main work on that first study I would like to present. Very dedicated and skillful PhD student funded by the China Scholarship Council. And what is depicted here is simply a pairwise matrix. The above diagonal gives all the SNP differences with this color scale, and the below diagonal the exonic non-synonymous ones. On average, two ramets, two modules that we sequenced have 2020 pairwise fixed SNPs. So what do we now mean with a fixed SNP? So since most of the genome at those locations is homozygous, so most of the allelic transitions, if we have a mutation that then manifests as a polymorphism within the ramets, go from homozygous to heterozygous. And we say that we have a fixed polymorphism if the frequency in the entire ramet that we've sequenced is 0.5, so it's heterozygously fixed in the entire tissue. And Lei used the GATK SNP calling pipeline, pretending that we had a recombining population, and he also implemented a number of hard filtering steps so as to be really sure that we only have true positive calls. And I should say that here, because that also means as a corollary that we probably underestimate the true amount of somatically generated polymorphism in our study, because we really wanted to exclude false positives. And also important is that we developed a restriction enzyme-based verification method. So sequencing independent, and we're able to verify a large proportion of the SNPs that we tested as a subsample, of course. We didn't test all the, in total 7,000 that were found. They then also constructed a neighbor joining tree based on that distance matrix that has a very high bootstrap support. And now let's have a look how the samples were really taken. So this entire bay is full of that seagrass clone here in Finland. And from ramet 1 to ramet 24, samples were taken at about 10-15 meter distance from one another. So the green ones are from the shallow area, the blue ones from the deep area. So from two rather contrasting marine habitats in terms of light, climate, and so on. We see that a lot of the genetic clustering of the genetically based topology mirrors the sampling location in space, but not all of it. So there are notable exceptions, and we believe that here probably shoots, ramets have been ripped off and have been rooted in somewhere else within the meadow, so that the topology is disturbed. Since we can safely assume that most of the transitions are from homozygous to the heterozygous state, we can also root the tree. So the root would be approximately here. And also bear in mind that the ramet 10, 8, and 12 will be later sequenced to ultra deep coverage to really go after the mosaic intra-ramet SNPs. I will come to that in a short while. So how can we be sure that this is really one clone and that not some sexual reproduction is hidden among those 24 sampled ramets? We believe that we can be as sure as it can get. If we here look at the heterozygote sites and how often they are shared among the ramets, we see over 30,000 that are identical and different to the backbone genome. And this very, very large number of genetic linkage, if you will, between 30,000 plus markers can only be explained by the absence of sexual reproduction. And Lei also ran a number of simulations, simulating some selfing among only 2 of the 24 ramets, and that would give a completely different pattern, spectral pattern of the number of shared heterozygote loci. On the other hand, we see here the somatically generated polymorphism, with the largest class of somatic mutations or somatic polymorphisms, I should say, so mutations that have been kept in the population and are now fixed, at the class of only one ramet. So they are essentially singletons, but that's exactly what you would expect under such a random mutation accumulation situation. And these are exactly those 7,000 we have just seen the slide before in the distance matrix. So how do these numbers compare to some other recent studies where essentially the same deep sequencing has been done, but not in a plant clone, but in a branching old grown tree, an oak? So in trees also there is the long-standing hypothesis that genetic heterogeneity accumulates in the branches that may also translate actually even to phenotypic differences, and that diversity within a single tree may differ or may create some resistance variation to herbivores or to plant disease. And some recent studies exactly address that in oak, but they found among very distant branches here that are about 100 years in terms of age distant from one another, they found very low number of SNPs, and they were mainly in a low frequency status, so not at F equal 0.5, so heterozygously fixed, but in a lower frequency, meaning mosaic status within the sequence tissue. In this study by Plomillon et al., it's interesting to note that 19 of those variants could be found back in the acorns, which is one of the first direct evidence that indeed, as it is also always stated, plants do not have a continual permanent germline, but essentially each leaf or each branch can differentiate into a flower, showing that somatically generated genetic diversity communicates with the germline genetic diversity in plants. So that's, of course, a very interesting additional issue, which I will touch at the very end of my talk. And so what's here now the similarity between an oak and a seagrass? So just think about a huge seagrass meadow as an oak in a two-dimensional space. So as if all of these branches and twigs would be cut off, they would have their own roots, and they would start growing in space almost infinitely. And yeah, we find among distant ramets within that seagrass genet, we find about 100 times more somatic genetic variation. And all of that I had been alluded to so far too is fixed. So it is present most probably in the entire plant module. And how is that possible then? Since we know from horticulture that many of the somatic polymorphisms are actually nice variations that look nice, and therefore they are marketed and sold, and they are persistent for a very long time. They are also called periclinal chimeras. Chimera is actually a bit unfortunate wording because a chimera means that you have a secondary fusion. So a mosaic would here be in a more appropriate name because it's an endogenous formation of genetic heterogeneity that manifests itself here also in bicolored leaf surface. Yeah, so how is it possible that we do find so many fixed SNPs, heterozygously fixed, most of them? So why are they not staying as a mosaic as in many other situations that I know? And to understand that, I would like to introduce the concept of somatic genetic drift, which I think is very important and that has so far been overlooked in the evolution of clonal organisms. And the key idea here is that if vegetative offspring is produced, it is always a subset of cells that make up or that are the founder cells for that new module, either through budding or through branching. So it is always a bottleneck, which means that the same basic principle as a normal genetic drift operate, and we can get the loss or the complete fixation of novel variation. So let me go through that concept a little bit more in detail. So I think nobody would deny that in any microbial population, if you have a favorable mutant arising here, that after a certain while, depending on the fitness coefficient, the selection coefficient, excuse me, so the relative fitness benefit and the population size and the time, this variant can rise to fixation. In a multicellular organism where you have tissues and the cells have their dedicated positions within tissues, yes, you can get a somatic variant, but then it basically sticks within that cell that sits in a certain tissue. You can get a second one down here, the red one, and all over again if you are an old organism like a human. So the human skin, for example, in people of 70 or 80 years of age is basically a mosaic of totally random mutations in the skin tissue, some of which may manifest, of course, into skin cancer, which we don't want to hope. But that's depicted here. So in unitary organisms, non-clonal, non-modular organisms, that's what I call unitary, like mice or men, if there's a proliferation of a mutation, then very often that's not a good situation. Then we may end up with a cancer tumor. But now enter modularity. So here with this sketch, what I try to show is that if we have only those two green mutant cells that form the new module, then through this daughter module formation, the genetic homogeneity would be restored in the new module. That's essentially another more graphical depiction of somatic genetic drift. So now let's get a little bit more natural, even here in the seagrass example. So here I've tried to depict this here in a growing seagrass ramet. So that's here the meristem, the budding, the proliferating zone of a plant that grows and from which all of the other cells, be it here the leaves or the shoot, are descendants. It's a relatively small zone of proliferating cells. And if a new variant emerges, we can have the situation that it's then mostly at the heterozygous state sitting in all of the tissue cells. So then F would be 0.5. The variant would be, as we also say, fixed. We can have the situation that it only sits here in 1 of 4. So it's diluted. That means that the allelic frequency is 0.0125. You will see in a minute why I go really into the number details here so that you understand the frequency spectra a little later. Or, of course, that's also entailed in the concept of drift. That new variant might as well be lost in another branching event in that module here. So just to show you what would happen if that mutation here in the mosaic state would rise a little bit more in frequency. Here, the frequency now would be 1/4 or 0.25. I had already alluded to the meristematic region in higher plants. Also, seagrasses have it. So if we understand the situation here and this kind of cell reactor, if you will, then we also understand the dynamic of somatic mutations within plant modules. So this meristematic zone probably has only in the order of 100 to a few hundred cells, maybe even less. We don't know yet. So that's ongoing investigations with histology, evidently. And so the situation would be simple if that would just be the cellular reactor, if you will, the budding zone. And then we could go on from there and study the conditions under which mosaicism or reciprocal fixation would happen. However, the complicating situation is that meristems in higher plants are often stratified. So they consist of layers where the cells have a pretty predetermined position and they are not interchangeable. We also say that it is stratified. And the layers probably also have a different size. So we have here 2 layers, L1 and L2, that are basically one cell layer thick, and then we have more a core zone here, L3, that is consisting of more cells. What would we expect if we now, to very high depth, sequence a plant, one module, in terms of all of the non-fixed mutants? So we would expect that we have a declining power law in terms of the variant read frequency distribution. The variant read frequency is what we actually measure in terms of Illumina reads, and that's our proxy for the true allelic frequency. If we sequence to a very high depth, which we did, 1,370, then it's a reasonable proxy for our true allelic frequency. Still, we do have, even with that exceedingly high sequencing power, we of course have a lower detection limit, which is in our case at f equals 0.008 approximately. The mutational input, as I said, is the lowest frequency class here. And then we have this decaying 1 divided by f square power law decrease. And so this is one of the real distributions that we found after that super high ultra deep sequencing. So two things that we did find is we do see here a low frequency peak, and then we see also here the decline actually, because we are here now at the detection limit. And we see the fixed SNPs that have just been shown at the very beginning in this distance matrix and in the neighbor joining tree. But we also do see these additional local maxima that are between our mutational input and the fixation for a heterozygote at 0.5. And they are, of course, very interesting, and they look like asexual sweeping cohorts of causal mutations dragging along, a lot of neutral mutations, but they probably aren't. So the more parsimonious explanation goes as follows. So if these are depictions of the relative abundance of the layers 1, 2, and 3 in that hypothetically structured meristem, and we would get here a mutation in the L2 layer or in the L3 layer, because the L2 layer only encompasses, let's say, a quarter of the total meristem. We would have an intermediate peak here once it is at fixation and drags along all the other neutral mutations in that background. And the same, and a slightly higher frequency peak would occur if variant sweeps to fixation in the L3 layer. So that, of course, complicates the picture quite a bit. What we also do see, and the peak at fixation here at 0.5 shows that under some circumstances, the designation to certain cells, to their layers, must be broken up. Otherwise, it would not be possible that we had mutations sweeping to fixation throughout the entire L1 through L3 layer. So this is something we have to postulate based on our molecular genetic data. We haven't shown that yet in terms of histology. We haven't even shown the layers. So these are just inferences that we think we can make because we see these particular patterns in the allelic frequency spectra. And again, of course, what we see here in that process would again be a different depiction of somatic genetic drift that breaks up, that goes across the stratified meristem. So the strongest evidence that Ley found in his work for somatic genetic drift is actually following those somatic genetic variants that were present in 3 of the ultra-deep sequenced ramets that are in that relative topology to one another. So R10 branches of the earliest, and R08 and R12 are more related to one another, so they should share more somatic genetic variation with one another than both to R10, which is actually exactly the case. And here depicted are about 40 somatic genetic variants, as I said, present in all of the three that are changing their frequency with some threshold that we've set. And we very nicely see that a bunch of them that are at an F of 0.2, they rise to fixation during that growth path here to both in R08 and in R12. Of course, there will be many that start off here with a low frequency that then are later lost, but they will simply not be depicted here because the condition to show this plot was exactly that all of those variants must be detectable in all of the three ultra deeply sequenced ramets, otherwise we would not consider them. So that picture in a way is biased. It only shows the upper half of the drift where low frequency variants increase over time as judged by the genealogy in other two ramets. So let me give you a quick summary. So what I've shown you is, first of all, that the pairwise difference in fixed SNPs is substantial within a genet, within a clone of a seagrass. So on average, we find about 2,000 somatic genetic variants, which is about in total 100 times more than in recent oak studies. To generate such a high number of heterozygously fixed SNPs, we think that somatic genetic drift, a previously unrecognized mechanism in clonal evolution, plays a key role. It essentially says that new branches are initiated by a subset of cells, and if you repeat that all over again through multiple branching events, through hundreds of branching events that must have happened, you end up with parts of that variation that is fixed, and parts of it, of course, is continually replenished in terms of mosaic variation. That brings me to the third point here. We do also find a lot of mosaic variation. We do find the mutational input peak, but we also find modes at frequencies below 0.5, below heterozygous fixation, and we believe that this exemplifies fixation in smaller tissue compartments in the stratified meristem that complicates the situation to be able to detect selective sweeps. So the bigger question then, of course, is after we've in principle have shown that there is a lot of variation, a lot of which changes even the gene sequences, and that we have a situation where not intra-organismal genetic heterogeneity would complicate the picture. So, but this genetic variation is fixed within the module that can be under selection. So we have an asexually evolving system under diploidy. What could possibly go wrong under those conditions? And actually a lot, at least according to conventional wisdom, could go wrong. First of all, under diploidy, the most, as I've shown, most of the emerging low frequency variants are heterozygotes. So they are changing a homozygous situation. So in order to create a phenotypic effect, they must at least be partially dominant in order to have any phenotypic effects. So in order to be seen by selection and diploids. Then secondly, under asexual reproduction, favorable mutations, they have to occur sequentially. So they cannot be recombined together in a relatively quick time in the same genotype if we had sexual reproduction. So may that waiting time for sequential favorable mutations too long? That's the big issue here. And thirdly, in terms of studying those systems, since the entire genome of an asexually propagating genet is a single linkage group, if you will, any causal beneficial mutation will drag along many, many other neutral variants, so it will be exceedingly difficult to tease apart the causal from the hitchhiking genetic variation. So let me go through those issues one by one. Mutations need to be partially dominant to be visible in diploids. So that's certainly true. But what's really the evidence that this has an appreciable effect on adaptation speed? And one of the best systems here is yeast, because in yeast, we have haploid and diploid forms that are asexual or sexual. So in this study here by Zeil, a similar study from 2003, haploids and diploids asexually were propagated over 2,000 generations, and there was essentially no difference in the accumulation of fitness. In the small populations, 10 to the power of 4, I should also mention here that in a large population setting, the haploids were faster, but nevertheless the diploids still had an appreciable adaptation speed. So this not necessarily seems to be an issue. In general, what one can say here is that the mutational spectra in terms of dominance, recessivity, they definitely need more study in those systems to really say how much the mutational effects would affect the adaptation speed. Second, the consecutive nature of mutations with and without recombination. With recombination, it's trivial. So if we start from an A-B situation here, so that's not one locus, these are actually two genes, and we have a beneficial mutation large B and another beneficial one large A. They can be brought together by recombination. That can happen relatively quickly. So they are united. Under asex, we have to wait within the clonal lineage large A, small b, so that the next mutation occurs, which would be capital B, and only then we have the required double, let's say, additive fitness effect of the capital A and capital B variant. So what's the evidence here that these situations, asex versus sex, have appreciable effects on rates? There are a number of experiments that do show appreciable effects on rates. However, the asexual populations also always do adapt, just a little bit slower. However, just a recent experiment to show you that this is by no means unequivocal is from Liu et al. In Nature, Ecology, Evolution. They had replicate diploid yeast strains. Some of them, the blue ones here, were asexual, the others were sexual, and they, over 1,500 generations, could not find any difference in the amount of adaptation and so in the rate of adaptation over that constant time period. However, the dynamics of which mutations then were retained in the adapting population, they were quite different in these asexual versus sexual diploid populations. So again, we can conclude that yes, we could expect in some circumstances that it's asexual diploid systems could adapt slower, but it need not to be so. So it's also not a killer criterion to say that those asexual systems cannot adapt with some reasonable rate. And now we can also come for the other end and ask, so what's actually the evidence from the phenotype that asexually clonal evolving systems do show some phenotypic variation that can be selected for? And maybe you all have probably tasted it already. And when I have my with you my Slack session this evening at 20:00 CEST, I will sip on it. So the white wine Pinot Gris and Pinot Blanc, they are actually somatically derived descendants by independent somatic mutations of Pinot Noir. And This paper here very nicely really breaks it down to the absolute genomic level, which color loci were affected. So these, so this phenotype that gives rise then eventually to white wine, which we can sip upon, is a purely somatically generated variant that has then been selected by humans for human consumption. Another totally unrelated example regards this invasive water plant here, Hydrilla, that's entirely clonal in North America, and 3 times independently, herbicide resistance was evolved in a diploid situation under in asexuality under clonal propagation. So let me now come to the third challenge, which is to study those systems. As I've said, the entire genome can be thought of an entirely linked linkage group, a humongous linkage group if you want, if you ignore the phase. And so if we now look at the among ramet level, so we're leaving the mosaic level, the polymorphism within the plant, but we go back to the fixed polymorphisms between the 24 shoots. Then we could have a situation like this. So a lot of neutral or almost neutral variants according to the neutral theory of molecular evolution hanging out there. And then we get suddenly one really good causal adaptive mutation that's under positive selection. So this ramet number 3 here would start proliferating over time and space, and it would drag along all the other neutral variants, of course. So they would also increase in frequency. They would look like they are under selection, but they aren't. They are hitchhiking. And you already see here that the key is to identify cohorts, modes of allelic frequencies that travel together in space and time. This should be the key of detecting those asexual selective sweeps. And that's also why the stratified meristem in plants makes this exercise exceedingly difficult if you already have some peaks for a completely different reason, which is the stratification and the separation of certain layers of the meristematic tissue. So therefore, for that selection analysis, we really would like to focus on the between ramet level. And where can we borrow knowledge and technology and approaches to model those asexual selective sweeps? So this is, I think, in cancer biology. So cancer, since about a good decade, is now viewed as a clonally evolving system. And I think that's very good because that provides a lot of insight, evolutionary insight into medical research. And here the cancer people have exactly the same issue. They have a few driving mutations that make those tumors explode and then later also metastasize. But they drag along a lot of other passenger mutations, as they also say, that are non-causal. And the trick is here also for tumor treatment, of course, or to assess whether some treatment had some success, to be able to tease those two sources of variance apart. And here Benjamin Werner comes into play, with which we would like to collaborate in the future a lot, since he has developed those cohort-like approaches where essentially the dynamics of a cancer progression in time here, so the cancer grows and grows and grows, is viewed, and then the same allelic frequency spectra that I've shown you a few slides ago are depicted for all mutations in those cancers. And what we would be like to look at are those, what the cancer people call subclones. So these are basically, in our case, ramets, sharing the same somatic genetic polymorphism and how they expand and how we can see them superimposed onto the neutral genetic, the neutral variation of growth of an asexual dividing system. So to adapt this a little bit more to the seagrass or to any other free living clonal system, so the null distribution would be like this. If all the ramets would just proliferate in space, and they would just accumulate somatic genetic variation. Here, this drop off here is again not a real biological phenomenon, but it's determined by our sequencing depth, which in turn determines the lowest variant frequency we can detect. And so if we now had a causal mutation here sitting in that ramet that makes it proliferate, then we would start seeing a subclonal cluster, or translated to the seagrass world, we would see a group of ramets sharing a certain group of polymorphisms that look very different to the neutral expectation here, bearing, of course, the fixation peak over here that occurs over time through somatic genetic drift, in combination, of course, with selection. Drift always occurs, but this is not to say that selection cannot make this drift pattern even more pronounced. Again, so in contrast to the other frequency spectra that I've shown you before, this is now the among ramet level of selection. So this is basically here the total population of ramets divided by the number of ramets, so plant modules that share a certain polymorphism. And that already tells you that to have a decent resolution, I need to sample a lot, which we will do with money from Human Frontiers of Science program. So as I've already introduced, Benjamin is on board to model cancer evolution and translate that to free living clone. And I'm also very happy to have Iliana Baums from Penn State on board. She will study the same phenomena that we found in seagrasses and corals, because corals are also modular basal animals. They also proliferate often asexually, form very large clones. And the same as in seagrasses, it is of a very high conservation concern to ask whether they can adapt by means of somatic genetic variation. So how would we do this? So we would really do a more fine-scale mapping, not just sample 24 of those plant ramets or coral colonies in space, but likely in the order of 100 or even more, depending on how we allocate our sequencing budget. This will, for the seagrass, again be done with Chris from Finland. And to show you already a glimpse of what we found among our 24 ramets, actually, this is an enlargement of the one slide I've shown you, while we are sure that we only have one clone. Remember, we had these large number of heterozygote loci, over 30,000, that were identical among the 24 modules. Here we now have the accumulation of variation in at the low frequency end. And here we see a little bump here that, of course, with a much higher sampling resolution, not just 24, but let's say 100, would maybe end up to show us one of those asexual selective sweeps. How does now the somatically generated variation compare to the variation owing to recombination? So here, this is essentially this neighbor joining tree when we scale the whole picture to encompass a sexually recombining combination from Sweden. So that's the nearest population we have from another different population genomic data set. So, and by and large, we can say that the pairwise SNP distance is about 7% of that distance, which would emerge if we had all of the genotyped plants emerging via recombination. However, all of this is de novo mutation, not just shuffling of existing variation. And all of that variation that manifests itself at the between ramet level also went through probably pretty drastic selective filters already. So it is not as unbiased as this variation that manifests itself after sexual recombination. What do I mean with here? What do I mean with this? Let's have a look here at a DNDS study that was done with all of the 7,000 interramet SNPs, and then the 3 data points of the ultra-deep sequenced ramets, which yielded always around 10,000 mosaic SNPs. So we see that whether they are only occurring in one ramet or also shared with other ramet, it doesn't really matter. So they seem to be under purifying selection. So the NDS is clearly smaller than 1. Whereas those that appear as fixed between the ramets, they are significantly larger and show a relaxed purifying selection. Since this is a composite measure, this can mean 2 things. It can mean that the purifying selection is has the same magnitude, but that it's just canceled out a little bit by positive selection. This we don't know, but as a matter of fact, we clearly see that the 2 groups of somatically generated polymorphisms, whether they are still mosaic or already fixed, they are under different selection regimes. And this is why we also think that we found evidence for multi-level selection here that is different among the 2 organizational levels, among cell lines within ramets and among ramets nested within a clone or genet. And this has been postulated already, or just last year actually, by a book by Michael Cruzan, Evolutionary Biology, a Plant Perspective. So in normal non-clonal plants, the number of long-lived plants, the number of deleterious mutations should slowly accumulate. So that's basically Malus ratchet here. And that should shift the distribution of the mutational effects clearly to deleterious, whereas it starts with neutral. However, if we have within ramet, what he calls here with intragenerational selection into play, then we have here this deselection, which I've just shown in the previous slide with the DNDS plot within the ramet, which effectively may mean that the mutational distribution stays around neutral, which among other things means that Malus ratchet would be very much delayed in a clonal plant. And yeah, whether this qualitative model turns out to be true, of course, we need to do more work here also on the actual fitness effects of the mutations and the polymorphisms that we do see. But I think it's a nice working hypothesis to have this conceptual model as a background. The final things I would like to allude to, and I actually see that I'm way over time already, is the germline concept by Weismann. So for plants, it's clear that the germline is transient, so that the tissue of the vegetative plant can reconfigure into flowers essentially. So that means that there is actually a feedback and contrast to Weismann back to the germline of processes in the soma. That's very important, and that's pretty much ignored in many evolution textbooks. For example, this seminal paper here is only cited 30 times since 1986, but I think it's a very important one because it exactly alludes to that point. Same may be true for basal metazones. So that's not at all clear if Weismann's doctrine really applies to those basal animals, such as corals, for example, or sponges, or other often marine things. Therefore, one of the objectives of Ileana in our ongoing project is to really check if the somatically generated genetic variation may end up in the coral gametes. So I end a little bit in a rush, I'm sorry for that, with the implications of our results for the definition of individuals. So 3 characteristics make up an individual. That's genetic uniqueness, genetic homogeneity, and physiological independence. We can make 2 ticks here, green ticks for uniqueness and independence. Homogeneity, of course, only always applies to those genetic differences that have become fixed, but all of the modules, they always carry a load, if you will, of mosaic variation around with them, but because that applies to each and all forms of multicellular life, I think we also need to relax that condition in general to define an individual, since everything, including humans, mice, whatsoever, are genetic mosaics. So the new view for clonal organisms, I think, that needs to be elaborated is that we have a nested system of individuals. So we have an asexually proliferating set of individuals that are really individuals that is nested within the genets that are also another different sort of individuals that are sexually reproducing, and both of those diversity levels, they communicate with one another. And with a thank you slide, I'm at my end. Again, I'm sorry that I'm over time, but I hope that I was able to entertain you, and I'm now happy to take on a few questions. And as I said, at 8 o'clock local German time, I'm happy to answer more questions on Slack. Thank you.
Fantastic. Thank you very much, Thorsten, for a very interesting talk. So while we're still getting questions starting to come in, I thought I'd start with a question of my own for you. So I was quite curious if you could speculate a little bit more on this idea. So, if somatic genetic variation within the whole clone between the ramets could contribute to adaptation, could you perhaps speculate whether different environments may perhaps influence the branching frequency in modular organisms to create more independent modules as one means of potential creation of genetic variation and adaptation?
The modules in the seagrass, they are independent already after 2 or 3 years. So essentially, they are not like a network like the hyphae in a fungus. So they are not really communicating. So we are really looking at asexual individuals already. Of course, the speed, I think what's really essential is not so much the mutation rate, but probably the branching rate, because that determines how quickly the mutations are brought to fixation, because the mosaic mutation essentially, I think, is just random noise. So I don't think that selection can really operate with a mix of different cell phenotypes here. So they need to be in the fixed status. And yeah, our branching rates are also very crude estimates. So we really need more a handle on what the branching rates are, and also what the average time, the average number of mitotic cell divisions is actually between any two branches, to also more calibrate the mutation rates.
Okay, that's great, thank you. So we have a question from our audience, which asks, do you have evidence of novel homozygote sites being generated by mitotic gene conversion or recombination subsequent to the new mutation?
That's a very good question. We actually have seen that in some of our phytoplankton evolving system, Emiliani-Huxleyi. They do sometimes mitotic recombination. We have no evidence found so far in our seagrass. So if we had that, we would see very different mutational frequency spectra. But that's absolutely an absolutely good question, because yeah, like I said, I mean, unicellular dividing things may do that, and then you get different mutational effects, of course.
Great, thank you. So I think we might have time for one more question for now. And then I think that the audience is still thinking out their questions as well, so we may be coming in after your talk. So yeah, it would be great if you have the chance to get back to any additional questions we have. So there's a question which asks, is there data on whether mutation rate among ramets is higher when environments change more?
No, we have absolutely no data on that at the moment. No, we don't, unfortunately.
Okay, that's great. Would you expect that it could be the case, or So hard to
Yeah,
tell.
I mean, we know that under stress, of course, mutation rates can be elevated, like UV stress, which is probably low in the marine environment. But of course, it's conceivable that other stressors are more important. The next question then, of course, would be, is mutation, is that the rate limiting step for adaptation, right? In microbial cultures, we do know that mutation isn't even often the rate limiting step to adaptation, right? So probably it is, but yeah, that definitely needs more work in our system.
That's great. Thank you. Interesting direction to pursue in future. Okay, I think we're stopped there, but yeah, we'd like to thank you once again for a very fascinating talk. So thank you, Thorsten.
Thank you for inviting me. Thanks.
Thank you very much. So just to remind everyone, our next talk will be from Professor Chris Wheat on the evolution of an alternative life history strategy in butterflies. So we hope that you can join us on Wednesday at the same time and same place. And in the meantime, check out our updates on Slack and our Twitter feed, spread the word about our seminars, and stay safe until next time.